So this is the biofluids collection training and for the VIVA Mind study. So during this training today, we will be covering GUIDs, the specimen collection schedule, the kit request module, specimen labels, handling and processing the specimens, sample shipping, the NCRAD website, and answering questions. So we'll have time for questions at the end, but also please feel free to jump in and ask any questions as you go along the way. Here is our general NCRAD contact information. This is our 800 number that will ring to all of the coordinators, our all study account that will email all of us as well, and then our website, which has a whole bunch of information and resources available for you. So let's first start with the GUID. We do request that you generate GUIDs for this study and record them on the sample form. The GUID is a subject ID that allows researchers to share data specific to a study participant without exposing any of their PHI. It's made up of random alphanumeric characters, and it doesn't include any of the PHI in the identifier. In order to generate a GUID, you will need an account at BRICS. And so you'll go to this website. This instruction is also in the manual of procedures, and you'll request an account. It can take a few days for your account to be approved, so you might want to keep that in mind. Once you have your account, then you go to the GUID tool and you create your GUID. You will need to have collected this information here from your participants in order to generate that GUID. This information, there's a little worksheet in the manual at the back of the manual to collect this information. It'll only be kept by you at the site. It's not any information that you will be giving to NCRAD or to ADCS. This is information for you at your site to generate this GUID. And the concept is that if you collect this information and generate a GUID for this subject at your site for your study, if the same exact subject goes to another site for another study, gives the same information because it's their information, and then when the GUID is generated at that site, they will get the same GUID generated for that project as well. So it's a good way to be able to know, like if we're doing QC and it looks like the same subject is in multiple studies, we can tell, oh yes, they really are, without having any of their PHI. So this is a summary of the biospecimens that will be sent to NCRAD. There are blood products and CSF that will be collected. At the screening visit, you will only be collecting CSF. At the baseline, weeks 4, 8, and 16, there will be blood products collected. At week 24, both blood and CSF, same thing with an early termination if you need to collect that as well. This is just a really quick overview. We will go into a lot more detail about all of these visits and collections as we go through the training. So the first is for the screening visit. At the screening visit, you will be collecting 15 mils of CSF total. Those will be put into 3 different types of tubes. We will go into this more in detail as well about the actual collection process later on, but it will be going into 3 different types of tubes. They will be placed into aliquots. Those aliquots then will be all shipped to NCRAD frozen, except for the one yellow cap cryovial that will be held at your site for local labs. Then at weeks 4, 8, and 16, the blood products will be collected only. It'll be 2 10 mil tubes, a 10 mil red top, and a 10 mil purple top. You will be aliquoting those into 1/2 mil aliquots, sending all of those frozen to NCRAD as well, and those will be shipped in batches. I guess one thing I should have mentioned for this is these will not be shipped in batches to NCRAD. They will be shipped more promptly because these are used for determining study eligibility. So we will go into that more specifically later as well, but please keep that in mind as we go through the training. So these will be shipped in batches and the screening visit will be shipped individually. Then the week 24 visit and the early termination visit are the same, And they are a combination of everything you're collecting at the screening visit and the other visits where you're getting blood products, CSF. So it's 2 10 ml tubes of blood again. It will be the red tops, the purple top, and 15 mils of CSF. All will be sent frozen to NICRAD, and these will also all be sent in batches. And again, the early termination visit is the same as the week 24 visit. So those were just meant to be a brief overview, and I promise we'll go into much more detail about all those components as we go along. So first, let's talk about the kit request module and how you will request kits for the VIVA Mind study. This is the link to the kit request module. This is in your manual, and there is a page on the NICRAD website under Tools for Active Studies for the VIVA Mind study, and there's a link to the kit request module there as well. So once you go into the kit request module, you will be able to request the blood collection kits or the CSF kits. So for blood collection, again, it's baseline weeks 4, 8, 16, 24, and early termination. The CSF kits are for screening, weeks 24 or early termination. Then there are 2 different types of LP kits in there. There's the 22 gauge and the 24 gauge. We have blood supplemental kits and CSF supplemental kits. Those kits are provided at the beginning of the study, but if you need to request another one later on, those are available to request. Those kits have just kind of an assortment of extra supplies in case you have issues with any tubes or any of the particular supplies along the way. As you're collecting other samples, then you just have some extras on hand to pull from. Then there's 2 different kinds of frozen shipping kits for this project. There are batch shippers, which are for the majority of the visits. And then there's the individual shippers for that screening CSF visit, because those do need to come to NICRAD promptly after they are collected so they can be used for enrollment criteria. Then if you need individual supplies, those can also be requested on there as well. We do ask that in advance of you starting your study that you please request initial stock of kits so we can make sure that those can be delivered to you before your start date. So this is just a little preview of what the kit request module looks like. Here is the site information. We pre-populate this with the information from your site. We sent out an email a couple weeks ago to gather this from you, and we've heard from most of you already with this information. If you haven't sent it yet, please go ahead and send that to myself and to Deontay, and we will be glad to get that populated in there. Then once you choose your site and that information is populated, then we'll ask you to confirm that the contact name, the shipping address, and the email address are all still correct. If anything needs to be updated, you can simply click no, that name is no longer the right contact person, and then it'll give you a box where you can provide the updated information. You can also let us know if it's, oh, just this one shipment needs to be updated because the usual contact is out on leave for a bit, or you can tell us this is going forward that you'll be the new contact and we should update our system to have your information going forward in the dropdown. So once you confirm your contact information, then you will be able to start selecting the kits. You can choose the number of kits in all different categories. So you can put a number for the baseline kits, you can put a number for the blood collection kits, whatever you need. Once you put a number into the box, then the contents show up at the bottom of the page, and that'll help you make sure that as just a visual double check, oh yes, that is the kit I meant to request, that those are the supplies I was looking for, and that's what I needed. That's also a good way to remember, oh yes, this blood collection kit doesn't have the frozen shippers. I also need to make sure I select the frozen shippers. So once you have everything selected for the kits that you need, Then you can also let us know if you need any other individual supplies. If you do need individual supplies, then you check the box for what you need. And then in the little comments box down here, you can tell us how many of each of those items that you need. Because there's not a place for you to tell us how many boxes or how many labels or things like that that you need us to send us otherwise. Once you get all that part completed, there's also another box here for comments, and typical comments we see are things like, I will be out of the office next week, so if you could please send me this shipment when I get back, then we know to go ahead and get it ready, but hold it for shipping until after you return. Or sometimes, and we do get those as well that say, oh goodness, I realized I have a participant coming in 3 days. Can you please rush and get this out right away? Those aren't our favorite. We ask that you please don't do that too often, but we understand that it does happen from time to time. And so that's a place for you to tell us that as well. Once you click submit, then an email is sent to us here at NCRAD to know that there is a kit request received. We go in and we check it out, and we'll send you an email to let you know we did receive your kit request. If we have any questions about the kit request, like for example, you didn't request any frozen shippers, then we might send you an email and just double check that you meant to leave those off. Maybe you had a bunch left over from previous request, or if it was just an oversight, that's a good way for us to just confirm you'll have everything you need when we send you your supplies. So just some friendly reminders that each site is responsible for ordering their own kits and making sure they have supplies on site for their scheduled participants. We don't know when your subject visits are, so please make sure you have everything on hand in advance. Again, here's the link to order the kits. Please visit that kit request module to make sure you have an understanding of how it works and are familiar with it before you before you need it. So I recommend you go check that link out. And then also please remember, it takes us about 2 weeks for your order to be processed. So please try to give us 2 weeks notice for your kit request. And then we can make sure we get it to you on time, especially now since COVID has happened. There are different issues that have happened with supplies and challenges with getting certain supplies in. So we stay on top of it as best we can, but we have limitations that we have no control over. So please try not to wait till the very last second to put in your kit requests, because it will take around 2 weeks in most cases. Any questions about the kit request module before we move on to the specimen labels? Okay, so let's go into labels. So there are 3 types of labels that you will use for the VIVA Mind study: the kit number label, the site and ADCS label, and the aliquot tube label. The one thing to note about the aliquot tube label is that it will differ by specimen type, and we'll show some examples of all of these. All labels are provided in the kits, so you will have everything you need when we send you the kits. Here's an example of the kit number label. So it has a barcode on it and this 6-digit number. These are used to track patient samples and help with providing quality assurance. They're placed on all blood collection tubes, the biological sample and notification form, and the CSF sample and notification form. For those visits where you're collecting both blood products and CSF products, you will have a different kit number on the blood product form and on the CSF products. So the blood samples and the CSF sample will have different kit numbers from each other, but all the blood samples will have the same kit number and all the CSF samples will have the same kit number within a visit. On the outside of the microcentrifuge boxes as well, you will also put a kit number label on those too, and we'll show a picture of that a little later on. The site and ADCS ID label looks like this. You'll see there are some blanks on here, and that's for you to handwrite in your site number and the participant ID. So since you do have to handwrite this in, we will provide a 2-pack of fine point markers in advance of the study starting that you can use to write on there. If you happen to lose those, you can request more in the kit request module, or they're just a fine point Sharpie marker in case you want to just grab something on hand before you can get any more ordered. We ask that you please write this information on the label prior to sticking them onto the tubes. It's a lot easier to write on a flat surface than on a round surface. And then these labels will be placed on the 2 collection tubes, the 10 ml lavender top EDTA and the 10 ml red top serum tube. The final label type is the aliquot tube label. And this label has the study name. So it says VIVA Mind. It'll have the sample type on it. So it'll say plasma, serum, buffy coat, or CSF, and it will have the kit number as well. Here are what the blood collection tubes look like once they're labeled. So they'll have 2 numbers on, or 2 labels on them, the kit number label and the site and ADCS ID label. Then the aliquot tube labels, it's this label will go on to this little cryo vial. I did want to note that these don't have a barcode on them because the cryo vials already have a barcode on them. So we are covered with the barcodes already on the labels, or on the tubes, on the cryo vials. When labeling the tubes, we ask that you please label all collection and aliquot tubes prior to cooling, collecting, processing, or freezing samples. This will help ensure that they adhere well to the tube. Please only label 1 subject's tubes at a time to avoid mix-ups. Even if you're seeing 2 subjects on the same day for the same visit type, we ask that you entirely label all of 1 subject's samples first before moving on to labeling the second subject's tubes. We ask that you wrap the label around the tube horizontally instead of vertically. So please make sure you orient them like this. And then it really helps once you put the label on to roll the tube between your fingers where the label is to really make sure that that label is stuck to the tube. Any questions about labels before we move on to processing the samples? Okay. So there is some equipment that we require that the sites already have on hand. The standard PPE, you need to have your own needles, a sharp spin, dry ice will need to be provided by the sites, and then a centrifuge that can spin at 2000 RCF and be refrigerated at 4 degrees. We will also need to store the samples in a -80 freezer until they are shipped. And at one part of the processing, the samples will be placed on wet ice. So these are the 2 tubes that will be collected. It's a 10 ml red top and a 10 ml lavender EDTA top. We're just collecting 1 of each at all the blood visits. It's really important to note that you'll collect the samples in the order of red top, then purple top. So you'll notice we always put everything in that order, red top, then purple top, just to help with that. There will be centrifuging required. And as I noted, it needs to cool to 4 degrees. And so it can take up to 30 minutes for that centrifuge to cool. So please plan accordingly and get that cooling prior to starting the blood collection so that you can have it cooled in time for processing the samples. So this, I think, is a really helpful slide in particular to kind of orient you to the different cryo vials and what they'll be used for. We really try to do a lot of color coding. It helps you, it helps us, and hopefully it all is pretty logical. So the 10 ml lavender top EDTA tubes will go into purple top cryo vials. So from a purple tube to a purple cryo vial. The red top serum tubes will go from the red top blood tube to the red top cryo vials. There are residual tubes that are blue capped, and that's for all sample types, they are blue capped for that residual. And we'll explain a little more about what a residual is a little bit later. The Buffy coat samples go into the gray cap cryovials. The CSF samples that come back to NCRAD go in the orange cap cryovials, and the yellow cap cryovial is for CSF to go to your local lab. There is an additional tube that we will collect CSF directly into, and it's a Sarstedt 2 ml tube that will be used for a Roche assay. And so we'll describe that a little bit here in the CSF collection section. So the first tube we're collecting for the blood visits are the serum tube. It's the 10 ml red top serum tube. So prior to the visit, you will want to store the tubes at room temperature and make sure the tubes are pre-labeled. Then you will collect the blood into the 10 ml EDTA and allow the blood to flow and ensure the blood has stopped flowing when you go to when it's all filled. Then immediately after the blood draw, invert the tube 5 times to mix the samples. After you have inverted the tube 5 times, then you'll need to allow the blood to clot for 30 minutes. You will then centrifuge the samples at 2000 G for 10 minutes at 4 degrees Celsius. The serum samples will need to be spun, aliquoted, and in the freezer within 2 hours from the time of collection, so please keep that in mind. Sometimes it can take a little bit longer than 30 minutes for the serum to clot, and that's okay, so just kind of keep an eye on it, but also keep that end time point of 2 hours in mind. There are videos on our website that can show you what the serum clotted looks like, so if you have any questions about that, feel free to check those videos out. Once you've spun the serum, then you will aliquot that into the pre-labeled cryovials. They will be aliquoted into 1/2 ml cryovials. And in theory, you should get about 5 mils of serum from a 10 ml blood draw. So that would ideally be about 10 1/2 ml aliquots. In reality, that usually doesn't work out quite so precisely. So if you have one that's a little less than a 1/2 ml, then we ask you to put that into the blue cap residual tube so that we know, okay, that one is a little bit less than the others that are at 1/2 ml. And then you will note that the last 4 digits of that barcode on the sample notification form as well, so that we have lots of ways of realizing that one particular tube doesn't have a 1/2 ml volume in it. Once all these aliquots are frozen, it's really hard for us to determine the exact volume in each of the tubes. So then this is just another visual of once the serum tube is spun, here's the serum that you'll be pulling off, and these are the RBCs and the white blood cells and the platelet clot that you'll leave behind in the tube. Here are the cryovials that you'll be placing in the cryo box. And that's it for serum. Any questions about serum before we move to the lavender top EDTA tubes? Okay, so for the 10 ml lavender top EDTA, you will, same thing, store the blood tubes at room temperature and pre-label the tubes prior to the draw and the visit. Then you will collect the blood, try to fill it all the way up, and allow the blood to flow for 10 seconds and ensure the blood is stopped. Then after the blood draw, immediately invert 8 to 10 times this time to mix the samples. Once you've done that inversion, go ahead and place the samples on wet ice until you can get the centrifugation going. Preferably within 30 minutes of the blood draw. We do ask that you centrifuge the samples at 2000 G again at 4 degrees for 10 minutes, same as the centrifugation for the serum. And just the same as the serum, the samples need to be spun, aliquoted, and in the freezer within 2 hours from the time of collection. These will also be sub-aliquoted into 1/2 ml aliquots. And ideally, again, you would get 10 1/2 ml aliquots but in reality, you might get one of those residuals again. Same thing here, go ahead and put that residual in a blue cap cryo vial and record those last 4 digits of that residual onto the sample form. And go ahead and stick all these plasma samples into the -80. After you get all the plasma aliquoted off, the next layer will be the buffy coat layer. So you will go ahead and pull that off and put that into the gray capped cryo vial. Once you have that, you'll go ahead and stick that into the -80 as well. And this is just another picture kind of giving you a visual of here's the plasma, here's that little buffy coat layer, and then you'll leave behind the RBCs. Just a cartoon of it as well. You go ahead and draw off the plasma, put all those into the purple top cryo vials, put that into the cryo box and add your buffy coat in there as well. And I think this is a really helpful picture for the buffy coat, because one of the really key things with the buffy coat is when you're drawing off that buffy coat layer, you will get some of the RBCs that are below the buffy coat, and we expect to see that. So when we get that buffy coat, we expect it to be definitely pink, if not red. And that's a way we know that not only was that buffy coat, or that all of that buffy coat layer is pulled into there as well. Because if you don't get any of that pink or red color to it, then we worry that it's still just plasma layer and not enough of the buffy coat is in there to get a good DNA extraction. So that buffy coat goes into the gray cap cryo vial and sent on frozen with the other blood products. Any questions about the blood processing before we go to the CSF collection? Okay. Well, I guess I should say, so this is what the LP tray will look like that you'll get. We don't require fasting for the LP. However, we do ask that you try to have the LP done on the participant at the same time of day each time they do have an LP. So if you did it at first thing in the morning, like an 8 a.m. or 9 a.m. visit, for that screening visit, when you do their week 24 or early termination visit, please try to do it the same time again for that visit. So 15 mils of CSF will be collected in total. Please pre-label all of the orange cap cryo vials and the yellow cap cryo vials. There will be an individually wrapped tube that you will not pre-label, and individually wrapped conicals that you will not pre-label either. Those are all individually wrapped because they are primary collection tubes and will be going into the sterile field. So we don't want you to pre-label those. Those will get labeled after the collection. So go ahead and pre-label those orange cap cryo vials though, and stick them in some wet ice to be nice and chilled while you're doing all these other steps. So you will do the lumbar puncture, and the first couple of mils will go into the 15 mil conical. If they are bloody, you'll discard them. If they're clear, go ahead and send them on to the local lab for local lab testing. After you take care of getting the clear collection and sending off some CSF for your local lab, the next collection you will do is to collect directly into this 2 mil Sarstet tube. So this is a primary tube, so don't collect into a conical. So you will collect directly from the LP into this tube. You will collect 2 mils into that. There is a fill line on there. You need to fill it all the way to that fill line, then cap it, label it, and set it aside to store. You won't do anything else to that tube. It's just fill it, label it, and freeze it. Once that 2 mils are collected, then you will go ahead and go back to your other 15 mil conical and go ahead and collect the remaining of the 15 mils of CSF. So it kind of depends on how much you've already collected here, but it's probably going to be in the neighborhood of about another 10 mils, 12 mils or so that you'll have to collect into this 15 mil conical. Go ahead and collect that in there. put it in wet ice until you're able to centrifuge that conical. Ideally, within 15 minutes, you will be able to get that centrifugation going. Again, it's going to be 2000 G at 4 degrees for 10 minutes. Once you centrifuge that conical, there'll be a debris pellet at the bottom. You're going to leave that and you're going to aliquot off all of the CSF into half mil aliquots into those orange cap cryo vials. If you have anything less than half mil, put it in the residual again. And just like with the blood products, you'll put it in a blue cap residual tube. You will record those last 4 digits on the CSF sample and notification form, and then put all of these in the freezer. So these will all go in a cryo box, which we'll show you in a minute. This tube does not fit in a standard cryo box, so we'll be storing it a little bit different. And this tube goes off to your local lab, the yellow. So any questions about collection before we go into shipping and packaging of the samples? Okay. So all of these samples are going to be frozen and shipped to NCRAD frozen. So please only ship Monday through Wednesdays. We want to do that to ensure that there are no courier delays that will cause shipments to be held at a hub over the weekend and dry ice to be melted and samples to be thawed. So please make sure you ship Monday through Wednesday. The screening CSF should be held no longer than shipped every other week. So if you are collecting the screening CSF this week and you have no other participants scheduled this week or next week, just go ahead and right away ship that to us at NCRAD, because we have to get that sent out for the assays to be done, for the screening evaluation to be done. But if you're going to see a subject this week and another one in a couple days, you can go ahead and hold on and send those together, but the most we want them to be held is every other week. And then all the other visits, the week 24 CSF and all the blood visits will be batch shipped together. So these shippers will have no more than 6 cryo boxes in them. And the combination of visits will kind of just depend on which time point it is, but it's 6 cryo boxes per shipper. So if you accumulate 6 cryo boxes in a few weeks, go ahead and send those out. But if you have visits spaced out, then please don't hold on to samples for more than 3 months. Even if you don't have 6 cryo boxes after 3 months, we ask you to go ahead and get those sent to NCRAD after 3 months' time so we can have those accounted for. Okay, so this is back to that summary table, and just a reminder that everything is coming frozen. Everything should be shipped Monday through Wednesday. The screening CSF hold on to no more than bi-weekly, so please don't hold it more than every other week. And the other CSF, week 24 or early termination, can be included with batch shipping. And this is a good reminder of how many aliquots or tubes will be sent back to NCRAD for each sample type. This is what the blood products will look like in their cryo box. So all of the visits collect the same type of blood samples at baseline weeks 4, 8, 16, 24, or early termination. Here are your plasma aliquots, your buffy coat, and your serum aliquots. Here's your kit number label on the outside of your tube. And please, please, please only put 1 subject per box. I know there's some open spaces here, but in order to avoid mix-ups on any side, we ask only 1 subject per box. Here's what the CSF collections will look like in their box. So note that these are only the orange cap cryo vials. The 2 ml Sarstet tube does not fit into these boxes, so we'll go over how to package that one up next. But again, please only put 1 subject per box and put the CSF kit number label on the outside of this box as well. And note that this kit number is going to be different than this kit number. So then these cryo boxes will go into, the blood cryo box will go into a small biohazard bag that'll be included in the shipper. And the CSF cryo box will go into a large biohazard bag because the Sarstet 2 ml tube does not fit into these cryo boxes. It will need to go into the 50 ml conical that we provide. That conical then will go into the bubble wrap, and that will go into the large biohazard bag with the CSF. So all of these items are from the same subject at the same visit in one bag together. Only one subject per biohazard bag, please. For the dry ice, please put some dry ice at the bottom of the box. Then you will put your biohazard bags with the cryo boxes inside there. If it's the screening visits, you'll have 1 or 2 cryo boxes per shipper. If it's the other visits, you will have up to 6 cryo boxes per shipper. For the individual or for the screening visits, it'll be about 10 pounds of dry ice. And for the other visits, it'll be about 45 pounds of dry ice. So please plan accordingly. Another important note is that there are labels for the dry ice on the packaging. You do need to record the weight of dry ice in kilograms on the packaging. If that is not filled in, then the courier can reject the package. So for this study, we're using UPS as the courier. We have a video on our NCRAD website that helps walk through the program we use called ShipExec for UPS. We will ask you for your shipping contact information. We have that from a lot of you already, and we'll use that to fill in all your contact information already into the UPS system. So it'll all be pre-populated. It's a pretty user-friendly system. The first time might take a little bit to get set up, but then once you get set up, it should be really smooth sailing after that. But if you have any questions, definitely reach out to us. We ask that you please send the biological and CSF samples and shipment notification forms in advance of us receiving the samples. So the day you're shipping the sample, please send us those forms. Please send us the tracking number. This lets us know to be on the lookout for the package. And if it doesn't arrive, then we know to start looking for it and to start doing our research into where it is and what's going on. The beauty of us using UPS now, as well as we have a contract with UPS that we can request re-icing of packages as well. If something is really delayed, we can ask them to please re-ice the samples and hopefully not lose any samples in transit when there are any delays. For those sample forms, go ahead and email them to our All Study account. That way we make sure everybody has access to them in advance of the shipment. Of course, everybody doing shipping needs to have training on shipping. So we just provide a few resources for IATA shipping in case those are needed. Please feel free to check these out as needed. And in case it's helpful to know when doing that training, all of the samples that we're shipping will be shipped as UN 3373 biological substance category B. So that would be the training that you would want to do if you need to do training still. Just another statement again, we can't drive it home enough that the sample forms need to be emailed to NCRAD prior to the samples arriving at NCRAD. In addition to sending those in advance, please also include a copy of the form in the shippers. That's really helpful for us to just match everything up from what we had in advance to what arrived. Here's what those sample forms look like. They're pretty similar to the T2 forms if you were part of that study. I think a lot of you were. We will ask you to fill in the GUID. You can put your kit number barcode there. You'll check what visit it is, fill in the subject information here, put some date and times here, and then down here, it's a lot of information about the processing of the samples. We also ask if you collected CSF or not, because then that helps us know if we should be looking out for that second kit. And then the CSF sample form looks almost identical, but just has fields related to CSF collection and processing. Okay, before we go into some typical non-conformances or issues that we see and some of our suggestions for how to avoid them, any questions on shipping? Okay. So let's go into these non-conformances. So just to try to get ahead of the curve, here are some typical issues that we see over time with different studies. And so we just try to be proactive and suggest some ways to not have them occur before the studies even start. So one of the things we do see sometimes are that sample aliquots and collection tubes can be frozen at an angle or completely upside down. We do ask that you please freeze them upright and keep them upright until the shipment. As I noted before, once they freeze, they're pretty hard for us to tell the volume in them, and they're even harder to tell if they're frozen at an angle or upside down and everything's in the lid. So it's really important to please freeze them upright for us. We also ask that you please complete the sample forms during the participant visit and as samples are processed. I know there's a lot going on at that time, so it's definitely a busy moment, but if you leave it and say, oh, I'll just fill it out later when things settle down, then often fields can be missed or left blank unintentionally because that information wasn't collected. The other thing is that we really highlighted that residual volume. We only want one tube per sample type that has less than that 1/2 ml in it. Please make sure that all the other aliquots are at the 1/2 ml. And in order to prevent multiples that are less than 1/2 ml, go ahead and lay out all the cryovials in a row and just aliquot in order until you run out of volume. We also would really like to stress that it's really important to ship all the frozen samples for one participant in one shipment box from the same visit. So if it's one of those like week 24 visits where they have both CSF and blood products collected, please put those in the same frozen shipper. So that's why we don't necessarily say to send the boxes from certain number of visits together or a certain number of subjects. We say send 6 cryo boxes because there could be just a blood sample if it's like a week 4 visit, or if it's a week 24 visit, there could be the blood products and the CSF products. So please keep that in mind when you're packaging the samples. Please make sure all aliquots are labeled. You'd be surprised how many times we receive unlabeled tubes, even though we provide all the labels in the kits. And then also please provide the forms in advance of the shipment. So, on our NCRAD website, in addition to having a VIVA Mind page, there are some other helpful pages. For example, there is a page that highlights our holiday closures. There's one for what to do for Friday blood draws, but that's not as helpful for this study because all the samples are shipped frozen in batches on Monday through Wednesday. So Fridays aren't as big of an issue. There is a VIVA Mind active study page. It looks like this, and here's the link to it. So I feel confident that all of you will become very familiar with this page and spend a lot of time here. There's a link to the kit request module here. We'll have links to the sample forms. After the last training, I did have a couple of updates to the manual procedures that were highlighted. So I have those done, and I'll just give it a day or two after this training to make sure no other questions or items come to light, and then we'll get a new version of the manual out and I'll get that posted on the website. We'll have this video posted here in the training slides. We'll have a copy of these slides posted, and then we'll also have some video tutorials in here, things related to drawing off the Buffy coat and things like that will also be posted in there eventually, and how to do some shipping and packaging as well. So that's all the information that I have. It's a lot of information, but if anybody has any questions, please feel free to reach out to us. Don't ever hesitate to ask us questions. We would much rather answer questions than have you struggling or feel confused or lost. We answer lots and lots of questions, so please always feel like you can reach out to us. Here's our general contact information where you can always get a hold of somebody, and if we don't answer right away, we will find the person that can answer the question for you. So anybody have any questions?